Okay, section 3, which is all about plant nutrition and transport. So the most part of this that you kind of need to be aware about, biggest chunk, is really photosynthesis. So you will know the word equation and the symbol equation for photosynthesis. So plants take in carbon dioxide and water, and they need light energy, so i.e. sunlight, or it could be an artificial source of light, and they produce glucose and oxygen. You need to know the symbol equation, which is kind of underneath the written equation. You guys need to remember that. It produces food in plants. We put food in inverted commas. Sort of like that. What it actually does is the plant produces glucose, and then the plant uses that glucose for respiration. So we would get our glucose from food that we eat. Plants can't do that, so instead they do photosynthesis to produce the glucose. It happens in the leaves of all green plants, so this is really what your green leaves are actually for, is for this site of photosynthesis. It happens in the chloroplasts. So remember, chloroplasts are the cell structure, the cell organelle that we've spoken about. And the chloroplasts contain a pigment called chlorophyll. The job of the chlorophyll is to absorb the sunlight, and then it uses the energy from that sunlight to convert carbon dioxide and water into glucose, as well as oxygen. Oxygen is just a byproduct for a plant photosynthesis. It's not what it's there for. The whole point of photosynthesis is to produce glucose. But obviously it's useful for us, really, because of all this oxygen production. Photosynthesis is important to plants because it converts that light energy into chemical energy, which is stored in the glucose. And then the chemical energy is released when the glucose is broken down during respiration. Now, it's worth knowing that producing glucose is the aim of photosynthesis, but respiration isn't the only use for that glucose. So plants will obviously use some glucose for respiration. Some of it will be used to help make cellulose, which they use to make their cell walls. Some of it will be stored as starch, some of it will be stored as lipids and fats, and then obviously the rest of it will go to that respiration process. Okay, so there are other uses for the kind of glucose that's made in photosynthesis. Now, limiting factors affect the rate of photosynthesis. We did a lot of work on this kind of at the beginning of the school year. So a limiting factor is something which stops photosynthesis from happening any faster. Okay, so it can't get any quicker because of this limiting factor. Now, your limiting factors for photosynthesis typically are light intensity, carbon dioxide concentration, and temperature. So the limiting factor depends on the environmental conditions. So for example, in winter, it could be your low temperature that's your limiting factor, whereas at night time, obviously sunlight is likely to be your limiting factor. So in more recent work, we've kind of looked at maximising crop yields, and we've spoken about, how a farmer might manipulate their greenhouse. What they're considering are those limiting factors. Now, there are 3 graphs that you guys need to know the shape of and be able to explain them. The first of which is when we have rate of photosynthesis against light intensity. So this is this graph at the bottom of this first page on photosynthesis. So you can see at first, as we increase the light intensity, the rate of photosynthesis also increases. Okay, but then it reaches a certain point and our graph begins to flatten out, it plateaus, it becomes that straight horizontal line. At that point, as soon as your line flattens out, light intensity is no longer a limiting factor because we're carrying on increasing light intensity, but you can see our rate of photosynthesis isn't changing. So the second that line plateaus, flattens out, light intensity is not the limiting factor, something else is, and that something else is going to be the carbon dioxide or the temperature. Okay, you won't know which, but it's going to have to be one of those two. Okay, the graph for carbon dioxide is exactly the same. Okay, it's one of the raw materials needed for photosynthesis, so it's one of the ingredients. So it makes sense that as you increase the carbon dioxide concentration, your reaction is going to happen more quickly because there's more ingredients, more raw materials, more reactants for it to kind of carry on. So similar shape graph as I'll flick back, as our light intensity one. You can see as we increase our carbon dioxide concentration, our rate of photosynthesis increases until that line flattens out again. Remember what that line flattening out tells you? carbon dioxide is no longer a limiting factor, something else is, and that something else is going to be either your temperature or your light intensity. All right, so carbon dioxide and light intensity graphs are exactly the same shape. Your temperature one is slightly different, okay? Now you need to remember that photosynthesis is an enzyme controlled reaction. So what that means is it has to have enzymes helping it conduct. So think back to what you know about enzymes. This is where having a good solid understanding of enzymes is so important, because they come up in digestion and they come up in photosynthesis. You guys know that temperature can either make the enzymes work more quickly, but once you reach that optimum temperature, that best temperature, anything hotter than that slows down your rate of reaction because the enzymes are denatured. And this is exactly what your temperature and rate of photosynthesis graph shows you. So initially, as we increase our temperature, our rate of photosynthesis increases, okay, because your substrate and your enzymes have both got more energy, so they're more likely to successfully collide, and therefore there's more likely to be a reaction occurring. So we've increased our temperature, our rate of photosynthesis has risen. Then suddenly you can see there's almost this kind of peak where you have your optimum temperature, and then our rate of photosynthesis rapidly decreases, actually down to 0, so to no reaction happening at all. And this is because our enzymes are being denatured. Okay, that graph is annotated with the phrase destroyed. We should be using the word denatured. Okay, typically though, as you can see, the temperature for denaturing your enzymes is quite high. You know, it looks to be maybe 50 degrees Celsius, something like that. All right, that is obviously quite high. So what happens more commonly is actually the temperature is too low. That's what happens more often than not. So, you know, it's a little bit low, so actually we could increase our temperature. Okay. Now, the leaf is kind of the main site of photosynthesis, and funnily enough, the leaf is therefore adapted for photosynthesis. So it's designed in a way to kind of maximise that rate of photosynthesis, to make sure it's happening as quickly as possible. You need to be able to label the leaf, so the diagram is there. I'm not going to talk you through the diagram. You can kind of look at it for yourselves and label the parts, but you do need to be able to pick them out. Okay, so you need to be able to say, label them if you were to be given a diagram. And then there are a few different adaptations that are worth knowing about. So leaves themselves are typically broad, so they're quite wide. So you're giving yourself a large surface area to expose to light. If all your leaves are really, really, really thin, then you're obviously going to need a lot more of them than if you had really, really broad ones. Most chloroplasts are in that palisade layer, so that palisade mesophyll layer, which is kind of located near the top of the leaf. So they're there because that's where they can get the most sunlight. The upper epidermis, so that very, very thin, very, very top layer, is transparent, so it's see-through, so light can pass through it easily to the palisade layer. Leaves have a network of vascular bundles, and within those vascular bundles are your xylem and your phloem, and we'll talk about those in a little bit more detail in a moment. But they help to deliver water and other nutrients to every part of the leaf and take away the glucose produced by photosynthesis. So your xylem your xylem brings the water up from the roots to the leaf so that it can use it for photosynthesis, and your phloem takes the glucose away from the leaf and delivers it to all the other areas in the plant that need it. You've got a waxy cuticle, and that helps to reduce water loss by evaporation. So again, when it's hot, you're kind of keeping as much of the water as possible in the leaf. And the plant has adaptations for efficient gas exchange, which again we'll look at in more detail in a later topic. But it has these little holes, they're stomata, and they allow carbon dioxide to diffuse directly into the leaf. So a couple of additional details that aren't present in this revision guide but are worth you guys knowing. Talking about your stomata, you've got the guard cells either side. They can help control the size of that stomata, the size of that pore, that hole. Okay, so if there's low carbon dioxide concentrations, the stomata would kind of open to maximise the amount that can get in. If it's a really, really hot day, they might close down a little bit to try and stop the water evaporating. All right, so the guard cells can help control the size of the stomata in terms of, maximising carbon dioxide concentrations or minimising water loss. The spongy mesophyll layer, which is kind of the one just over the stomata, you can see is labelled with, is labelled with an airspace. Those airspaces help with the diffusion of gases through. So having airspaces through your spongy mesophyll layer means that gases can diffuse through more easily and kind of get to the places they need to be, which is the chloroplasts. Now, there are a few investigations that we can do in order to kind of measure photosynthesis and, you know, check that it's happening, prove that it's happening, and measure the rate of photosynthesis. I'm actually, because this is quite long and quite wordy, I'm going to do this in a new video. All right, so join me for part 2 when we're going to talk through these photosynthesis experiments.